Now, the chromosome theory of inheritance basically states that Mendelian genes, these genes that Mendel described, they have specific loci or locations on the chromosomes. And in addition to that, the chromosomes do follow segregation and independent assortment, which were Mendel's laws of inheritance. So if we look back at meiosis and throw in a couple of Mendel's genes as well, the genes are located on chromosomes. Here we have two pairs of chromosomes. We have a larger chromosome that happens to contain the R gene, and we have a smaller chromosome which contains the Y gene. When the parents undergo their meiosis, they are going to produce gametes that each have basically one chromosome from each of those homologous pairs. If those combine in the F1, we ended up with heterozygous F1 individuals. Now here, when we look at the next meiosis that's going to take place with those F1 hybrids, in metaphase 1, those homologous chromosomes can line up in different ways. So the tetrads may line up so that both of the red chromosomes are on one side, or they may happen to line up with those red chromosomes on opposite sides of the metaphase plate. That's the law of independent assortment. Now, when we get to anaphase 1, the homologous pairs of chromosomes separate from each other. So notice here that the capital R is moving one direction and the lowercase r is moving another direction. That is the law of segregation. So they do separate from each other. In the end, when we do have the law of segregation and the law of independent assortment, when we're working with two different genes, as we are on this slide, we do end up with 4 possible gametes from that dihybrid individual. That's exactly what we looked at in the last chapter. Now, Thomas Hunt Morgan, he did a lot of genetics work as well, so he continued a lot of Mendel's experiments, except that he worked with fruit flies. Now, he had a lot of good reasons for working with fruit flies. Many of them were similar reasons to why Mendel was working with his garden peas. First off, they bred at a very high rate, and they had a very quick generation time. So he was able to produce a new generation every 2 weeks. And another very important thing for him was that they have only 4 pairs of chromosomes. That is quite a small number of chromosomes. And in his case, he was really trying to prove that the genes that Mendel talked about were indeed located on the chromosomes. So in this case, he did not have very many chromosomes that he really had to sift through when looking for his gene locations. Now, he had a population of fruit flies, and pretty much all of his fruit flies had a red eye color, as you see in this picture. That red eye color, because that is the normal version that the flies have, that is what we designate wild type. So you can think of wild type as being the normal version. Some people will call it the native version of the gene, but that's what the fruit flies have. Now, he came across one fruit fly that had white eyes instead of red eyes. So that was his mutant. It was important because that gave him something that he could look at how it was inherited in the offspring. So he had this eye color gene, 2 versions, red versus white. Now, genes are typically named for whatever the mutant looks like. So in this case, the mutant has white eyes. So we have a W representing the gene. And then when we're talking about the wild type version, we put a little plus next to it, as you see over here. And that designates that that's the wild type normal version of the gene. So what Morgan did was he took this wide-eyed fruit fly, which happened to be male, and here is our symbol for male if you aren't familiar with this. It was a male. So he took that male and he crossed it with one of his normal red-eyed female fruit flies, and he looked at the offspring. So he took a look at the F1 generation, and what he found was that every single one of the F1 offspring had red eyes. So that was pretty similar to what Mendel got in his experiments. It basically told him that red was dominant. And he then said, okay, well, let's take a couple of the F1 individuals and let's cross them and produce the F2 generation. When he produced the F2 generation, he got a 3-to-1 phenotypic ratio. So again, very similar in that case to what Mendel had gotten. But the interesting thing here was not only did he have a 3-to-1 ratio of the phenotypes, but all of the white-eyed fruit flies that he got happened to be male. So he did not have any white-eyed females in this F2 generation. So this indicated to him that somehow the eye color was related to the sex of the fruit fly, such that at this point in his experiments, all he had seen was white-eyed fruit flies. At this time in history, there were microscopes available. Microscopes could be used to visualize the chromosomes inside the fruit flies. So Morgan already knew that there was a chromosome difference between the female fruit flies and the red-eyed fruit flies. Well, as it turned out, this eye color gene happens to be located on the sex chromosomes. Specifically, it's on the X chromosome. And if we look at this cross, if we take our female right here, so this will be our female, she has 2 X's. Okay, because fruit flies are going to be very similar to animals or humans in that regard, where the female has 2 X's. And then the male we're going to have over here, and the male has an X, and he also has a Y. This eye color gene is located on the X chromosome. So this means that for a female is actually going to have 2 alleles for eye color, and the male, since it only has 1 X, is only going to have 1 allele for eye color. Well, the female, she was wild type, and she was a homozygous red-eyed fruit fly, so we want to put 2 W pluses on her X chromosomes. On the male that we're crossing it with, he had the white version, so the regular W. So these are the parents that we start with. This would be what's called the P generation, or the parental generation. And from this parent, we're always going to have an XW plus contributed to the offspring. So if we draw ourselves a little Punnett square here, that is the only option from our female. So we can put an XW plus, and then our male could contribute either his X, which has a regular W, or his Y. When we fill out the boxes of this Punnett square, now we can see what combination of W's they have, and we can also see whether they are male or female. So this box right here would represent a red-eyed female, and this box over here would represent a red male. Okay, and remember that in the F1, which is what we're looking at here, all of the offspring, male or female, did have red eyes. Well, what Morgan did next was he took those 2 individuals there and he crossed them with each other to produce the F2. Now this time when we cross them, this female, she can contribute either one of her X's. So if we take those and we put them on one side of the Punnett square, she can contribute her XW plus or her X regular. And from the male, can contribute either his XW plus or a Y. So now this time when we fill out the boxes, if you look at what we get, we've got first, in the first box, we've got a homozygous red female. Under that, we have a heterozygous female, but she happens to have red eyes as well. We've got a red-eyed male, and we've got a white-eyed male. So that's our F2 generation, and that is our 3-to-1 ratio that we're looking at right there in the boxes. Okay, so this one would be red, this one's red, this one's red, and this one down here is going to have the white eyes. So that is the way that we work out crosses involving genes that happen to be located on the X chromosome. Now, Morgan had not seen any white-eyed females. That's not to say that white-eyed females in fruit flies can never happen, but for a female to have white eyes, she would have to actually have 2 W's that have 2 of the X chromosomes with the regular white-eyed W on them. That's going to be less likely than for a male to actually have the white eyes. So what we're looking at here now are what's called sex-linked genes. Sex, male or female, is an inherited phenotypic character. So we do call it a phenotype because when we think male or we think female, we're not thinking X and Y chromosomes. We have specific looks, appearances, things that we associate with that. Now, it is indeed controlled by the sex chromosome, so there is a genetic basis there. If you look in this picture here, this is a picture of the human X and Y chromosome. Notice that they look very different from each other, and these would not by any means be considered homologous to each other. When we talked about homologous chromosomes before, homologous chromosomes we said needed to be the same size and shape, so they needed to look the same. These do not look the same whatsoever, and if they were going to be homologous, they would need to contain the same genes on them. These do not contain the same genes, which is why we only had the eye color in fruit flies located on the X chromosome. We didn't want to put one of those on the Y chromosome because the Y chromosome has completely different information on it. That's also a good place to point out that we can have genes that have absolutely nothing to do with sex determination on the X and the Y chromosome. Eye color is a very good example of that. We'll look at some other examples along the way. So although some of the genes are involved in hormones, many of them are not. Now, if we look at the XY system, which is how sex is determined in humans and the vast majority of animals, basically we have 46 chromosomes. Of those 46 chromosomes, 44 of them are going to be what we call the autosomes. So hopefully this is a little bit of a review to you. So we have 44 autosomes, plus we have 2 sex chromosomes. So the X and the Y's are sex chromosomes in humans. So males and females both have 44 autosomes, and then a male will have an X and a Y, and the female will have 2 X's. When the male undergoes meiosis to produce the sperm will get 22 autosomes and either an X chromosome or a Y chromosome. When we have meiosis for egg production in the female, she will produce eggs that have 22 autosomes plus 1 X chromosome. Now, the female will always contribute an X to her offspring, and then the male will contribute either an X or a Y, 50% chance of either one, and that's why typically we say that the male determines the sex of the offspring, because he gives either an X or a Y. If he contributes an X, then it will be a female offspring. If he contributes a Y, then it will be a male offspring. You do want to be familiar with the XY system if you're not already. You want to know how many autosomes we would have in a typical somatic cell versus how many autosomes we would have in a gamete, and then also how many sex chromosomes we would expect in each one of those. Now, there are other ways to determine sex. They're not all X and Y systems. So if we just point out a few other options. In birds and some insects, we have what's called the ZW system. The ZW system is not so different from the X and the Y system. It's just kind of flip-flopped. So in this case, it is the male that actually gets 2 of the same chromosome, and the female is the one that has the 2 different chromosomes. Another way that sex is determined is the X0 system. This is in grasshoppers and cockroaches. In this case, we have only 1 sex chromosome. It's an X. And basically individuals get 1 copy of that, or they get 2 copies of that. So in this case, you have males and females that actually have a slightly different chromosome number from each other. And then the last possibility we have listed here, Haploid Diploid Ii. This is something that we have in some colony insects like ants and bees. And in this case, there are no actual sex chromosomes, but instead sex is determined by ploidy. So the males are haploid, and the females are going to be diploid. So there again, we have a different total number of chromosomes in our males versus our females. Now, all of these examples on here, you do not have to memorize all the specifics of them, but you do want to understand that the sex does have a chromosomal basis. That is what's determining the sex of the offspring, either specific sex chromosomes or numbers of chromosomes that are causing that to be determined. We do want to point out, though, that there are a few exceptions to the rule where sex is determined by some environmental conditions during egg incubation. And this is something that does apply to a number of reptiles. So things like temperatures during egg incubation can shift the population towards male or females. Now, any gene that happens to be located on either sex chromosome is what we would call a sex-linked gene. And keep in mind, as we said, those can be things that have absolutely nothing to do with sex or male versus female, hormones, things of that nature. There are genes that would be located on the X chromosome. Those we would specifically call X-linked genes. And then there are genes on the Y chromosome, of course. Those would be called Y-linked genes. There's not a lot of genes on the Y chromosome, so we're really going to be focusing on the ones on the X chromosome. So these will be X-linked genes for us. These genes are going to follow specific patterns of inheritance because now we have a different number of alleles in our males versus our females. So in our females, we have different possibilities for the genotypes in that we can have homozygous dominant, homozygous recessive, and we can have heterozygous. When we're talking about the males, they only have one version there. So they may have the dominant version, they may have the recessive version. We just look at a couple Punnett squares here just to give you an idea. In this case, we are crossing a homozygous dominant female with a recessive male. Now notice we're just calling him a recessive male because we can't really say homozygous because he only has one copy there. In this case, we would get all dominant looking offspring. If we were to switch it up a little bit and we have a heterozygous female crossed with a dominant male, now we have 3 parts dominant looking offspring and 1 part recessive offspring. If we switch it up again and we have our heterozygous female crossed with, say, a recessive male, now we've got some recessive looking males and female offspring. Notice here that we can't really ever use the term heterozygous when we're talking about our males, or homozygous for that matter. Instead, we tend to call the males hemizygous. And what that term hemizygous refers to is that they only have one allele present there. So there is no homozygous, there is no heterozygous. And if we are talking about disorders located on the X chromosome, and there are some genetic disorders located on the X chromosome, the males are not going to be able to be carriers for those particular disorders. Because if you keep in mind what we talked about at the end of chapter 14, a carrier is someone that does not have the disease, but they have the allele. They have to be heterozygous. We cannot have that condition when we're talking about something that's hemizygous or only has one version. Okay, so if you have a sex-linked trait that is due to a recessive allele, the females will only express the phenotype or only look recessive if they are homozygous. So they would have to have 2 copies of the little a, for example. So here we see that written. If the females have at least 1 copy of the capital version, then they're going to look dominant. Now the males, on the other hand, they will express the recessive phenotype with only 1 copy. So if a male looks recessive, you know for sure that this would be his genotype. If he looks dominant, then this would be his genotype. So it is easier to figure out the genotypes of the males because their phenotype shows you whatever allele they happen to have on their X chromosome, where with the females, you still have the same issue that we had in the last chapter, where if they're recessive, then it would be like this. But if they're dominant, we don't really know what's in that second position. It could be a capital A, it could be a lowercase a. If we look at some examples of human diseases that are caused by X-linked recessive alleles, we do have a number of examples. Probably the most common of all of the examples is red-green color blindness. Notice that this is in 8% of males. So that's quite a lot of males. It's pretty common to have this particular type of color blindness. It is just one type. And then it's in 0.5% of females. So 0.5% of females have it, 8% of males have it. This is typically what you're going to find when we're talking about X-linked recessive disorders in humans. It's going to be more common in the males because they only have to have 1 copy of that particular allele and they will show that phenotype. The females, on the other hand, will have to get 2 copies of the allele before they would show the phenotype. However, if you think about these males that have it, the males inherit an X and a Y. If they inherit an X and a Y, keep in mind that they got their X from their father, they got their Y from their father, which means that their X, which was the one that had the red-green color blindness allele on it, that came from their mother. So for all of those 8%, they are inheriting it from their mother, but probably most often their mothers are going to be heterozygous, so they would be considered carriers for that particular disorder. The next one on the list is muscular dystrophy, a type of muscular dystrophy. We've got our percentage there for males. It is so uncommon in females that we don't even have a real recorded percentage there. Hemophilia, which is a blood clotting disorder, we can see that the female percentage is much lower than what we have in the males. And then in the last one, Rett syndrome, we would expect the males to have a higher percentage there. The problem or the reason that it's not shown there is because in the males it is lethal. So those are not males that actually survive. So just to emphasize, if you look at this picture here, when we're talking about X-linked recessive disorders, the mothers, the females, they can be carriers. However, the males cannot be. The males either have the disorder or they do not have the disorder. So we're not going to have the situation where you've got a heterozygous male that has it and he doesn't know he has it and then he passes it on to his offspring. If we look at an example here, we said red-green color blindness is caused by a recessive mutation of a gene on the X chromosome. So in this scenario, if we suppose that we have a normal man, so by normal we mean non-colorblind, and we have a woman who is a carrier for color blindness, they're going to have children. And now we want to know, what is the probability that their children, regardless of their sex, so regardless of whether they're male or female, what's the probability that they're going to be colorblind? Well, just as before, the first thing we need to do here is we need to figure out the genotypes of the parents. My suggestion would be that if we're dealing with something on the X chromosome, we go ahead and we give the female or the mother 2 X's, and we go ahead and we give the male an X and a Y. Then we can actually put our letters onto those X's. So we've got a normal man. Because he is normal, he has the capital version. So he has a capital A. We want to make sure that we do not put an A or an allele for this gene on the Y chromosome. The Y chromosome in these crosses is really just kind of serving as a placeholder for us. If we look at the female, she's heterozygous, so she must have a big A and a lowercase a. And now if we simply do the cross, we've got either one of the X's passed on from the mother. The father is contributing an X with a big A or his Y. So if we fill in the boxes here, we can see that this bottom box would be the only offspring that we would expect to be colorblind. So the probability that their children, regardless of sex, would be colorblind would be 1/4. Now, if we were to change the question a little bit and simply say, what is the probability that they have a son, or if we say consider they had a son, what is the probability that their son will be colorblind? Well, in this case, we would just look at the sons, which is right over here. And if they have a son, the probability that he will be colorblind is basically 1/2. So notice how the probability changes a little bit if we say regardless of sex or assuming that they have a son or assuming that they have a daughter. You want to make sure that you can work out crosses involving sex-linked genes. You do have a number of practice examples on your practice questions on Angel. So do go through and take a look at those. Now, X inactivation is kind of an interesting phenomena that happens in females. If you consider that females have 2 X's and males have only 1 X, and you also consider that a lot of the information on the X chromosome has absolutely nothing to do with sex, females have kind of double the information compared to what they really need. So they really just need the same quantity that a male has. So the process of X inactivation is kind of nature's way of leveling that dosage out. So getting the female's kind of working number of copies down to 1, just like the male's working number of copies. So what this means is that of the 2 X's that are within a female cell, one of them will be randomly inactivated during embryonic development. So very early during development, it's going to be shut down. Now this shutting down or inactivating is basically compacting it or condensing it And if we condense it or highly wrap it around some proteins, that means that we're not going to be able to access the information that's there very well. The information is still there, but it's really hard to get to. So if we look at this example here we have a calico cat, and calico cats are a good visual example for X inactivation, but that's not to say that it is the only place or that this is the only gene that has X inactivation applying to it. Hair color in cats is controlled by genes located on the X chromosome. When you have a calico cat that is a female cat, most often, you may have a few rare exceptions to that where you have a male calico cat, but we're talking about a female because she has two X's. She is heterozygous, she has one copy of an orange gene, and she has one copy of a black gene. So in this case, very early during development, when she has basically just a pile of cells, each one of those cells is going to randomly choose to condense or turn off one of the two X's that they have. When that X is turned off, it is highly compacted or condensed into a short, fat little chromosome, which is called a bar body. So what that means is if we look at this cell up here, the chromosome containing the black copy is shut down, so we are working off of the orange copy. So any cells where that happen to look orange. Some cells are going to randomly choose off the other one, which means that they are working off the black copy, and so those cells are going to look black. So when we see kind of the modeled appearance of a calico cat, we're seeing which cells decided to turn off which X chromosome. This happens in female cells because female cells have the two X's to work with. Should not be happening in a male cell because a male cell should not have an extra X chromosome to turn off. So every single cell always needs one working copy of the X chromosome. If you happen to have an odd situation where you had a cell that had 5 X's in it, then we would expect to find 4 bar bodies in there, because if we've got 5, we're going to keep one as a working copy, and we're going to shut down or compact the other 4 chromosomes.